this. Yeah, so I'm going to have to ask you to be understanding with me here. I've never used Microsoft Teams before. At Plattsburgh State, we use Zoom, and I know how to do that, but we're not doing Zoom here, so. Oh, right here, this one. Okay, I'm going to start out here in Brightspace. Can you see my Brightspace page? Can you let me know if you can see my Brightspace page? Yes?
Yeah, I can see it.
You can see it? Okay, good. Yeah, sometimes that's the confusing thing when, like, I'm looking at a screen and I'm talking about it, but no one else can see it but me. So just keep me up to date on that. So let me look at the introduction real quick. Some of you probably read it already. Okay, so just a little introduction to myself first. I am a professor over at Plattsburgh State. Been there for 30 years teaching cell biology. Before that, I did my PhD in University of Cincinnati School of Medicine, and most of my friends were med students. And then I went to Harvard Medical School, and I was teaching medical students there. So I have a good sense of what medical students want, what they need, how to teach this class in that respect. So, but uh, let's see. So basically, I'll be talking about a lot of the same stuff I do as for undergrads, except I want to go into more depth with you people, maybe even going a little bit faster because, when you get to med school, it's like they drive a dump truck of knowledge up to your head every day and dump it all in. So you have to get used to learning things quickly and taking in a lot of information. My undergrads go really, really slow, and that. Hello, can you hear me? Can anybody hear me?
Yeah, we can hear you.
Yep.
Wow, struggling today. Sorry about that. Yeah, my camera's not working for some reason, so you unfortunately can't see what an incredibly good-looking guy I am, but we will get back to how good-looking I am at the next lecture, I hope. Okay, so let's see if I can't get back to introduction here. Okay, can you see my PowerPoint?
Yes.
Yep.
Okay. Wow, what a mess this is. I wish I could just do Zoom. It's so much easier. But anyway, hopefully we're back on track now. So first slide here, this is a joke I always play on my undergrads. This is what you learn about cell biology as a high school student. The nucleus is the brain of the cell. The mitochondria, as we all know, is the powerhouse. The Golgi is the post office. The Refiar is the factory of the cell. So hopefully we're going to do things in a little more depth than that in this class. We'll see. The first slide here, this is just a picture from your book of a membrane, what membranes are like. So we're going to talk about membranes first. Now I'd like to start on slide 3. Now that I've looked at them closer, I think that's where I want to start. You probably all know that membranes are made of phospholipids. And this is kind of how we always think of them, a big round ball with 2 legs coming out of it. So this big round ball is a polar head. So you've got a big phosphate group there. Remember, phosphate is negatively charged. It's always big and ugly. It really sticks out. And then some polar group attached to it. And then you've got 2 fatty acids that are very nonpolar sticking out from them. And the basis in the middle is glycerol. Excuse me. We'll see that structure in a second. So the nice thing about phospholipids is if you get enough of them together, the heads are going to interact with each other and the tails will interact with each other. So you put a bunch of these together, they will form a micelle, which is just a kind of a little tiny sphere with all the fatty tails in the center. So this is just a monolayer of micelle. Interesting thing about micelles, they're starting to use them for drug delivery lately, which is really cool. I mean, if you've got a drug that's very non-polar, how do you get it into the body? One way is to put it inside a micelle, and then your body will absorb it like it would any other fat, and then the drug can get into your body. With these phospholipids, again, if you under the right conditions, usually right ionic conditions, plus you vibrate the solution, they'll form a bilayer. And this is how we see it in plasma membranes and internal membranes. All membranes in the cell are a bilayer. And then again, under certain conditions, you can form them into a liposome. So a liposome is a bilayer that forms a small sphere. The liposome is kind of analogous to a vesicle. A vesicle will look like this too, although vesicles tend to be a little bigger than what we see here. And again, these liposomes are often used for drug delivery as well. So if we look at the structure of, let's see, nope, that didn't work. If we look at the structure of phospholipids, here is your glycerol backbone. I can't even see this, it's so small. How do I make this bigger? Good view. Nope. Boom, 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 boom. Okay, it's a little better. So anyway, your phospholipid, the big thing is the backbone. The backbone is glycerol. So glycerol is just 3 carbons with an OH off each carbon, and you've got something esterified to each of those alcohol groups. Typically, you've got a fatty acid on the first 2, and then a phosphate and some polar head group on that last one. In terms of what the polar head group is, 45 to 55% of the phospholipids in membranes in mammalian cells will be phosphatidylcholine. So this is choline. And you probably know choline is also used as part of a neurotransmitter, acetylcholine. So this would just be added to an acetate, and that's acetylcholine. So phosphatidylcholine is approximately 45 to 50% of the membrane. Keep in mind in this class, I'm never going to ask you to memorize 45 to 50%. Just know choline is the major constituent. Then you've got phosphatidylserine, which is going to be important as a signaling kind of molecule. We'll see that in a minute here. Phosphatidylethynolamine is there at about 10 to 25%. And then you've got phosphatidylinositol. Now, this is relatively small in terms of percentage, 5 to 10%, extremely important for signal transduction. We'll see a lot of different signal transductions that happen based on phosphatidylinositol. So keep that in mind. So inositol is basically just cyclohexane, right? 6 carbons in a ring with an OH coming off of each ring. And one of those OHs is esterified to this phosphate. So this is like a phosphodiester bond, like we see in the backbone of DNA is also phosphodiester. What else? So some of the other things you'll hear about, phosphatidic acid. If you cut the polar head group off, you're left with just 2 fatty acids on glycerol and a phosphate. That's phosphatidic acid. And here's something that comes into play from time to time, lysophosphatidic acid. So if you take one of those groups off, the second group, you end up with lysophosphatidic acid. So make sure you know the names of all these things, and they will come into play. So here's the figure from your book. So here's one thing I wanted to say in the introduction. I'm not crazy about the figures in your book. The content is good. For the most part, I like the way it's written, but the figures I'm not crazy about. So I'll be adding my own figures from time to time. And if it's this figure's from your book, if it's not from your book, I will reference it so you know where it came from. But anyway, here's your glycerol backbone here. And here it's esterified to a fatty acid, and there should be another fatty acid here. So here's your ester bond, if you remember from organic chemistry. You've got a carbon, double bond oxygen, single bond oxygen, carbon. So what's really important and interesting about this ester bond is that it's really easy to take apart and put back together. It's like Legos, snapping things together and taking them apart. Because you've got this carbon here, which has got a very electronegative oxygen here and electronegative oxygen here. So both those oxygens are pulling electrons away from the carbon, which makes the carbon very somewhat positive, right? Not a full positive charge, but somewhat positive, which makes it easy for water to come in and bond to that oxygen and break the bond. So ester bonds are used in a lot of places, and we'll see them come into play quite a bit. All right, and here's your phosphodiester over here. Can you see my arrow? Anybody?
Yes.
You can. Okay, thank you. So here's your phosphodiester. Again, phosphoester because it's phosphate double bond O. And then you've got an OR on either side of it. So that makes it a phosphodiester. What else? Sphingomyelin, kind of a big, ugly, lipid soluble thing. And another part of your membrane. I forget now, probably 5-10% of the membrane. I'm not going to talk about it very much. It doesn't come into play very often. So, but that's another constituent of the membrane. Another thing I wanted to talk about was these fatty acids. They tend to be they can be anywhere from 14 to 24 carbons long. And when you look at membranes in a mammalian cell, they get thicker as you go from the rough ER to the Golgi to the plasma membrane. Plasma membrane is the thickest membrane, and the rough ER is the thinnest. So you can get a different length of fatty acids in the different membranes, and that it kind of helps to target proteins to the right membrane, because if it has to stay in the rough ER, then it will have shorter transmembrane regions to make it stay there, as opposed to something for the plasma membrane will have longer fatty acids. But typically in mammals, your fatty acids are between 16 and 20 carbons. 20 carbon with 2 unsaturations is arachidonic acid, and that's another one that's really important. Arachidonic acid tends to be in the second position, right here or right here. And it can often be cut off by the enzyme phospholipase A2 and made into leukotrienes and prostaglandins and local modulators. So these are basically paracrine hormones that go from one cell to another and activate signal transduction. Something on the first slide I forgot to say. Let's back up a little bit. We've got 2 enzymes that are going to be really important in terms of breaking down phospholipids. One is phospholipase C. That one's really important. That will cut on this side of that oxygen, releasing inositol, usually inositol trisphosphate or inositol diphosphate, triphosphate, yeah. That's really important for signal transduction. And the other one, which I just said, is phospholipase A2, which will cleave this off and release this fatty acid, which tends to be arachidonic acid. It'll then be converted into some signal transduction molecules. Okay. D, we don't really use D very often, so I'm not going to worry about that one. Okay. So those are our fatty acids. What else did I want to say? Okay, so that's it for fatty acids. The next thing I want to throw out here is cholesterol. So cholesterol is a really interesting molecule. It's amphipathic. It's got a polar group on one side, and then the rest of it's all non-polar. So that alcohol group is going to associate with the polar head, and the non-fatty parts, or the non-polar parts, are going to associate with the non-polar tails. Now, the reason cholesterol is important, it kind of acts like antifreeze in the radiator of a car. When they first started making cars, they would put water in the radiator. Well, water boils at 100 degrees and it freezes at 0 degrees, and you can experience, and that's just too narrow a range for the kind of temperatures an engine will experience. So they put glycerol in it. When they put glycerol in, now it freezes at minus 30 degrees and boils at 250 degrees. So now you've got a wider range to work in. Cholesterol is doing the same kind of a thing. If you look at this melting graph here, this blue line is if you don't have any cholesterol there. So basically, you've got a membrane that's very gel-like, very thick, until you hit a certain temperature, and then it starts to become fluid, and then it becomes really fluid. So basically, if you don't have any cholesterol there, the temperature of your cell has to be in a very narrow range. That's between these two. Um, you add cholesterol there, and now it's going to be more fluid at cold temperatures and less fluid at high temperatures. So it's modulating that fluidity of the membrane. And fluidity of the membrane is key. It's so important because the fluidity of the membrane is going to determine um, uh, the activity of membrane proteins. So if you're talking about the brain, right, it's all about membrane proteins, ion channels and action potentials and all this kind of stuff. A good example of what happens when you mess that up is inhalation anesthetics, right? Halothane, urethane, chloroform, ether. When you inhale that, it goes to your brain and kind of intercalates here between the phospholipid tails and makes it a lot looser, makes the membranes very fluid, and then your brain shuts down because when you change the fluidity of the membrane, the ion channels stop working and the nerves stop working. So fluidity is really, really important to modulate, and it needs to be within a certain range. And cholesterol makes that range a little bit wider, so it's really important for your membranes. Okay, lipid turnover. Now, the next slide is one from your book, and I'm not crazy about this. It doesn't really exemplify what I want to say. So let me just talk for a bit, and I'll come back to this figure. Basically, if you think about a cell and its membranes, usually we see a static picture in the textbook and we think the membrane is there. But in reality, it's all dynamic. There's always new lipids coming into one membrane and out of that membrane and over to other membranes. Everything's constantly changing the cell. And so this is how that happens. So in fact, if you think about it in terms of things like endocytosis and exocytosis, if a cell was constantly exocytosing something, like say a cell in the pancreas that's secreting digestive enzymes. Well, if you're constantly secreting, your membrane's going to get bigger and bigger and bigger, right? So you have to have a way of retrieving that membrane again to keep your plasma membrane stable. Same thing if you're endocytosing. If you're constantly pulling membrane in, your plasma membrane is going to get smaller. So these things are constantly turning over. You always have new vesicles with new phospholipids going into membranes and coming out of those membranes. And the membranes are made in the smooth ER, right? Smooth ER is responsible for making phospholipids and making new membranes. And then little vesicles will bud off of the smooth ER and go over and bud onto like the Golgi to add phosphates there, or the plasma membrane to put phosphates out, or phospholipids out there. So keep in mind that you've got a constant turnover of membranes within cells. My legs are getting sore. Okay, so fluidity. So your membrane phospholipids are not attached to anything as a rule, right? They can turn around in place. They can diffuse from one place to another. There's a lot of different movement that happens with your phospholipids. Another thing they can do is flip from one side to the other. So this phospholipid head can come down through the membrane and go to the other side, so they're flipping to the other membrane. Now that's the other leaf of the membrane, I should say. You've got your outer leaf and your inner leaf. That's rare, okay? It's possible, but it's rare. And you can imagine why that would be rare. You've got this big polar head group, and it has to go down into this lipid environment and then come out the other side. So it's not going to happen very often. But the cell's got ways of making these things flip. So when your phospholipids are made in the smooth ER, they'll butt off and then go out to the plasma membrane. And then in the plasma membrane, you've got enzymes like flipase, floppase, which is in here, and scramblase. And I always say to my undergrads, this is proof positive that scientists have a good sense of humor. So we've got flipase, floppase, and scramblase. So flipase flips them from the inside to the outside, floppase from the outside to the inside, scramblase does them in both directions. So the idea here is that Because of the activity of these enzymes, phosphatidylcholine is going to be the major constituent in the extracellular membrane. I mean, extracellular membrane, the outer leaf of the plasma membrane. And then you've got a lot of sphingomyelin exposed there, and then the inner leaf of your plasma membrane is going to be mostly phosphatidylethanolamine and phosphatidylserine. Now, earlier when I said phosphatidylserine, I said it's going to be important for signaling. This is the one example I know of where it's used for signaling. And the idea is phosphatidylserine should be on the inside of the membrane. If it's exposed to the outside of the membrane, macrophages are going to come along and say, this is a sick cell, let's destroy this cell. And then they signal it to go into apoptosis. So, and why is that? Well, phosphatidylserine tends to be on the inside, but if the cell is in distress and its ATP levels drop, as your ATP levels drop, calcium levels come up because you're constantly pumping calcium out of the cell, but you run out of ATP, that pump slows down, calcium increases, and your flop pace is going to stop and your scramblase is going to keep going, and then you end up with phosphatidylserine on the outside of the membrane. Here you can see it a little closer. And when this happens, if you block flipase and activate scramblase, you've got phosphatidylserine on the outside of the membrane. That's going to trigger apoptosis. So back up and review all this again. Basically, you've got a lot of membrane fluidity here. These phospholipids can go back and forth a lot. They tend not to flip or flop. They can, but it's very rare. But your cell is going to regulate what that outer leaf of the membrane is going to look like in terms of phospholipids. And when it loses that ability to regulate, it's about time to kill that cell. Okay, let me pause for a second here. Any questions? That's about all I'm going to say about the phospholipid parts. Any questions about this? No questions? Comments? Discussion? Okay, so let's go on. Oh, another thing I wanted to say about this class in general is, and I had so many internet problems to beginning, I lost my tempo there. I'm not sure. I originally planned to do 2 chapters per exam, but when I started reading this textbook, each chapter here has got an awful lot of information in it. This chapter 2, is basically 3 chapters, in my opinion. You've got lipid structure of the membrane, you've got transport across the membrane, and then you've got electrophysiology, really, membrane potentials and action potentials and things like that. So I'm not really sure how much is going to be on the next exam. I figure we'll have 2 lectures a week, 3 weeks, and at that point we'll negotiate between us where the next exam is going to be in terms of how much material. I do have exams scheduled. Someone asked me if I could make it a window instead of a time to take it. I think that's a good idea since it's online. So right now, I think the first exam is scheduled for February 5th. I'll probably change that to like February 4th to February 8th. So, you've all probably got jobs and things you need to do. This will give you the opportunity to fit it into a window instead of having to be somewhere at a specific time. So anyway, that was more intro that I didn't get to talk about earlier. But bottom line being, I'm not sure how much material I'm going to get through before the next exam. I may get like partway through the next chapter before we have an exam, but we'll talk about that more. And again, this is what I'm talking about in terms of feedback. How are you doing? Is this too much already? Do you think this is fine? So let's discuss. Let's have feedback. Okay, so next thing is membrane proteins. So membranes are all well and good, but they're pretty useless in terms of communicating with the outside world, right? All they are is structural, really. So everything that happens in a membrane is going to be because of membrane proteins. I always like to say proteins are like the hands of the cell. Anything that gets done by the cell gets done by proteins. Everything else is just storage or structure. So we've got a number of different types of membrane proteins. First type is a single pass protein. So this um, transmembrane region here, TMR, is an alpha helix that goes through the membrane. And your alpha helix is typically between 20 and 25 uh, hydrophobic amino acids, nonpolar amino acids. So your side chains are nonpolar. And I say 20 to 25 because, if you go back to what I said earlier, your membranes are different thicknesses depending on where they are. So your rough ER is going to have your shorter transmembrane region proteins in them. And that helps it to stay there because it's not going to be as stable if it goes into a thicker membrane. And your plasma membrane is going to have the longer transmembrane region. So your single pass protein then has got a single transmembrane region, which is 20 to 25 amino acids long. It's got an extracellular domain, and if it's going to be in the plasma membrane, typically that extracellular domain will have some glycosylation on it. That's what this orange hexagon is. It's one sugar basically, but you'll have like chains of sugars coming off of it. And that's really important for identification by the immune system. So the immune system's always going around looking at cells, making sure they're actually part of you and not invaders. And they're looking for these glycosylations to identify those cells. So your extracellular domain will typically have that. And then here you've got an intracellular domain as well, which other proteins can attach to. So that's your single pass protein. Uh, Most of your proteins, I believe, I may be making that up, but that's okay. I can make things up. Um, are going to be multi-pass proteins. So it goes in and out of the membrane multiple times. And in cartoons, we tend to show them this way, but in reality, they tend to bunch together um, to form one functional unit. So that's your multi-pass protein. And then you've got proteins that are attached to proteins that are transmembrane proteins. So these are going to stay with the membrane because they're bound tightly to these other proteins, but they're not actually in the membrane. Then you've got some peripheral proteins that are ionically bound to a phospholipid. So you've got part of what goes on in the Golgi apparatus is covalently attaching phospholipids to proteins, or even just fatty acids to proteins, and that'll keep them attached to the membrane. So that's this lipid attached peripheral protein, and then this protein is just bonded to either the head or has got some hydrophobic areas that make it stick into the membrane a bit. And then you've also got peripheral proteins that are attached to the particular head, the inositol. And we'll talk about an example here in a sec here. So here's just some example of these. Glycophorin is one we'll see in this chapter. Transmembrane protein in red blood cells, really important for attaching the cytoskeleton to the membrane. Here, a multi pass protein. The again in red blood cells, it's called band 3. Band 3. Um, it is a chloride bicarbonate antiporter, which means that chloride comes in, bicarbonate goes out. And that's extremely important for transporting oxygen through the blood because, the carbon dioxide in tissues can get converted into into bicarbonate, and then that bicarbonate will be transported out for chloride coming in. That's going to make uh, the cytoplasm more acidic, and that's going to cause the oxygen to come off the red blood cells better. I'm not sure we're going to get into that whole mechanism here, but keep in mind that this antiporter is really important to the red blood cell in terms of transporting oxygen. D and chirin is another protein we'll talk about involved in the cytoskeletal structure. Um, a Nexin is part of what do you call it? The spot wells, integrins, the focal adhesions, Annexin will be in focal adhesions. We'll talk about them in this chapter. RAS is a signal transduction molecule. It has a phospholipid attached to it, covalently attached to it, so RAS will always be on the membrane. And then here, acetylcholinesterase is the enzyme that breaks down acetylcholine in cholinergic synapses. We won't be talking about that for a while, but we will get to that eventually. But anyway, these are all the different types of membrane proteins that cells will have. What time is it? 11:44. Okay, so here's something I'd like you to read. In the book, there's a clinical case 2.1, sickle cell disease, symptoms, diagnosis, and treatment. And then also, I believe they go into malaria and heterozygote advantage. I'd like you to read that over. So here's something I'd really like to stress. One major problem with the African American community is people don't believe them when they're in pain. I mean, sickle cell anemia can be extremely painful because it's basically the red blood cells are sickle shaped and they can't get through the capillaries, and so you don't get oxygen to your tissues. So it's like your whole body turns into this lactic acid pain kind of a thing. And they're undertreated when they go to the emergency room because they think, oh, this person's just looking for drugs. Sickle cell anemia is so important in that community. So it's important to know as much as you can about it, because you will be seeing, when you become doctors, you will be seeing people with sickle cell. So keep an eye out, read that section over, and I'll probably ask you questions about it on the test. Okay, so that's it for membranes in general. We're going to go into membrane transport now. Any questions? Let me stop my share for a second. I don't know how. Put my camera on. Nope. Okay, stop sharing. Stop presenting. To switch presenters, select cancel. Okay, let's stop presenting. Make sure we didn't go off. Okay, can you guys turn your cameras on for a moment? I don't think I can turn mine on for some reason. Yeah, nobody's camera's on. It mustn't be working for some reason. All right, so anyway, at least put your mic on briefly and say hello. Let me know that you're following along fine here.
Hello.
Hi,
Zoe Alpert, how you doing?
I'm good. I'm sitting in the LaGuardia Airport right now.
Oh God, I wish I was there. Ha, not.
My flight was
Okay.
delayed 3 hours.
Oh my God. Because of snow or what?
Yeah, the snow.
Oh, bummer. Bummer. All right. So am I going too fast? I feel like I'm going fast. What do you guys think? Nobody's got an opinion here? I think what I'm going to do is, go ahead, Pat.
I was just gonna say, I think it's going good. I don't know if you could see the videos. There was a lot of thumbs up and nodding of the head, so I think it might be okay
Oh.
for some other people
Yeah.
as well.
I don't know why I can't see anybody's videos here. I have to figure all this software out because this is just not working the way it should be. But anyway, good. So I think I'm going to put up another survey after this class is over, just to check in and see how you guys are all doing. Again, feedback is key for me here because this is the first time I'm doing this class. Can you guys see me or no?
No, I
No.
can't.
No? Yeah, I keep clicking on camera, but it's not working, so I don't know. All right, well, let's go back and start membrane transport. We'll probably go through passive transport, and then I'll take a break, and then we'll pick up again on Thursday at 11. By the way, I checked out the survey that's up there already. So far, Monday, Wednesday at 11 is winning. So that's probably where we're going to be next week, but I'll put it up in your calendar so you don't miss it. Okay, let's get back to work. Okay, membrane transport. The most important thing to remember about membrane transport is energy. Because if the cell has doesn't have to use energy, it won't. So what I mean by that is like if the cell needs glucose and there's more glucose outside than inside, it's not going to use energy to ship that in. It's going to go in passively. On the other hand, if the cell needs to get something out of it against a concentration gradient, it's going to be using energy. So energy, as we know, because we all love biochemistry, delta G is equal to delta H minus T delta S. So H is heat, the heat energy. So if we need to add energy, we're going to use ATP, and that's where our delta H is coming from. And S is entropy. So if you've got the same concentration of a certain thing on either side, that's the most possible entropy, right? The most mixed up it can be. If you have more on the outside than on the inside, that's a decrease in entropy. So if we want to do that, if we want to decrease the entropy by making concentration gradient, we're going to have to add heat energy or ATP in order to get that to happen. So keep in mind the energy, that's what's most important here. In terms of spontaneous things, like anything minus delta G is spontaneous. To get that to happen, it just happens spontaneously. But we've got two different ways of that happening. One is simple diffusion, which is just diffusion straight through the membrane. And the other is facilitated diffusion, where you've got a protein in the membrane that's helping that thing get across. Now, if we want to produce a concentration gradient, we've got two different ways of doing that. One is by directly using ATP, and that's our pumps, your calcium pump pumping calcium out. And the other way is indirect active, where you've already got a concentration gradient of one thing, and you use that thing to bring the other thing against its concentration gradient. So you're concentrating thing two by letting thing one go down its concentration gradient. That's indirect active. And that's like your sodium glucose transporter. You've got a concentration of glucose outside that's higher than inside. Maybe you've got more glucose inside than outside. So you can use that glucose to bring whatever glucose is out in with it and increase that concentration gradient. So we'll talk about that later in this chapter. But here we are, Fick's law. So Fick's law tells us the rate at which something will diffuse into the cell. So here we're talking about some molecule, Y in this case. It has to go into the plasma membrane, dissolve into the plasma membrane before it can get through. And that idea there, that has to be in the membrane before it can go through, is going to affect what can come in and what can't. But anyway, Fick's law, basically the rate that something's going to come in comes down to its concentration gradient. The higher the concentration gradient, the faster it's going to come in. The A is the area of the membrane. I know we've got a microbiologist here. He knows really, really small cells have got a really high surface area to volume ratio. They can transport things really, really quickly. The larger the cell gets, the lower that goes. Here in this class, basically, most cells are going to be about the same size, so we're not going to change this at all. What's most important to us here is this, the permeability factor, which means how easily is it that this can get into that plasma membrane and then through. And there are two main things that are going to determine this permeability factor. Let me see if this animation is going to work first. Can you guys see this animation?
No.
No.
No?
No,
Okay.
but when I clicked on it, it did start to animate.
Okay, so if you click on that, if you click on that, you can see it on your own computer. But basically, it's showing the difference between a non-polar thing that can diffuse through, which means it can go anywhere, versus a polar thing that can't get in because it will hit that lipid area in the center and be repelled by it. So one of the things, important things in the permeability factor then is the, let's go again, the partition coefficient. Will the thing dissolve in water better or an oil better. So the oil-based things are going to be anything non-polar. Non-polar things can go through membranes easily. And that's key when we talk about drugs getting into your brain. You know, you've got that lipid blood-brain barrier. So good example of that is Imodium. Anyone ever hear of Imodium, the drug? Imodium, it's used for diarrhea. It stops your bowels from moving, basically. Why is that? It's a morphine derivative. It's an opioid, but people don't get high on it. Why? Because it's polar, and it can't go through membranes, can't get through the plasma, the blood-brain barrier into the brain, so you don't get high on Imodium. But anyway, bottom line, one of the important things in diffusion is the partition coefficient. If it can go into a lipid membrane, then it can diffuse through pretty easily. And even really large molecules like steroid molecules will go right through a membrane. If it's polar, then it really can't get through. And you can click on this later and you can see my animation about that. The other thing that's important in this P, this permeability factor, is the size of the molecule. Small molecules, because they're small and slippery and sneaky, they can slip right through the membrane, and so they're going to equilibrate across it. So anything like CO2 or oxygen, water is a really great example because water is extremely polar for the size of it. It's a very polar molecule, but it can slip right through the membrane because it's so small. There are easier ways of getting it through, which we'll talk about shortly. But in general, you know, osmosis is based on water just passing through the membrane, and we know how easily that happens. Ethanol is on the small side. It's also somewhat nonpolar. It can go right through the membrane. Although getting back to membrane fluidity, you drink enough of this ethanol, it makes all your membranes in your brain fluid, and that's when you really get sick and possibly die. Low dose alcohol will activate specific receptors. Higher dose alcohol change the fluidity of your membranes and then shut your brain down. And we want to avoid, once you get into med school, you realize the main thing we want to avoid is the brain shutting down. That tends to kill people. All right, larger polar molecules though, like glucose, can get through a little bit. That's like about the max size of a particle that can get through a membrane. That's like molecular weight of 180. That's about max. Now, if something is charged, it's not getting in no matter what. All right. And we know that things like this need to get in, right? We need glucose, we need amino acids and ions. So in a second, we'll talk about how those things get through. But in terms of our Fick's law, if we keep this constant and we vary this, then this is going to be the slope of the rate of how it gets in. So let's see that real quick. Okay, so again, Fick's equation here, if we is basically a linear equation, y equals mx plus b. B being 0 because when there's no concentration gradient, nothing's going to be moving. Here's your concentration gradient, which we're going to increase as we go this way. And permeability then will give us our rate. So if you compare a different molecule that has a greater P, this would be steeper. If it has a lower P, like glucose, it would be shallower over here. So the slope of your line here tells you what is the permeability of that molecule going through the membrane. But again, it's linear. Simple diffusion is just a linear process. It's all just based on, for any one particular molecule, it's all based on the concentration gradient. Okay, that's it for passive. All right, so this is passive too, osmosis. Osmosis is something I think that confuses students sometimes. The one rule to remember is that water will always follow the solute. So, and that's true in all membranes of your body, especially the kidneys, right? If your body wants to get rid of water, it's going to get rid of salt, and the water is going to go with it. So water will always follow the solute. So what is osmotic pressure? Basically, if you've got this tube and it's open to the atmosphere, so you've got the same pressure coming down on either side, one atmosphere. and then you put a salt solution on one side that's more concentrated than the salt solution on the other side, and a membrane in the middle that's permeable to water, but not to the salts. What's going to happen is water is going to go from the low concentration side to the high concentration side, and it's going to keep moving until the solute concentration is the same on both sides. And now the difference between atmospheric pressure and where this ends up, where this ends up is the osmotic pressure. How much did that osmosis of water moving to dissolve the solute over here push against the atmosphere in order to change the height of this solution in the tube? So that's osmosis, basically water following solute, and it's always going to make sure that the concentration is the same of all solute on either side of that semi-permeable membrane. So in terms of red blood cells, this is where red blood cells, like most, an awful lot of the plasma membrane stuff that we know about plasma membranes came from early experiments with red blood cells because they're easy to get to, right? You can easily get some red blood cells to work with. You don't have to kill an animal. You can just bleed yourself a little bit. So anyway, a lot of this was done with red blood cells. So in isotonic solution, iso means the same. So if you put a red blood cell in a solution where the number of solute on the outside is the same as what's normally inside a red blood cell, then you've got a net no movement of water, right? Net is the same, water moving in is the same as going out. That's isotonic, the cell is a normal shape. If you put it in a hypertonic solution, there's more solute outside than inside, then water is going to come out until the concentration of solute inside the red blood cell is the same as whatever solution you put it in, right? We're not going to change the concentration outside. We're going to change the concentration inside by water coming out. And then if you put it into a solution where it's hypotonic or undertonic, then you've got less solute outside than inside. So water is going to come in to dilute that solute, and the cell is going to swell up. And if it's too great, the plasma membrane is only so big. So if that concentration gradient is too great, then the cell is going to explode. You hear a little pop sometimes when they go off like that. Let's see, 286 milliosmolar is, I'm wrong. No, that's right. That's about normal osmolarity for a red blood cell, 286 milliosmolar. You go higher than that, you're going to shrivel. You go lower than that, you're going to swell. And if you go far enough, eventually burst. And this is an experiment from your book that's kind of interesting. You can use red blood cells as an osmometer, basically. You just put cells in different solutions. This is isotonic on the right over here. So the cells are still intact and they sink to the bottom of the solution. As you go into more hypotonic solutions, cells start to burst, and eventually they will all burst. So as you go to lower and lower osmolarity, cells will swell and eventually burst. And then you can see the solution's all red here because all the hemoglobin came out of the cells as they burst. I don't know how important this is, but it's in your book. Seems kind of interesting. Okay. All right. What time is it? 12:04. All right, I'm going to do facilitated diffusion, then we're going to call it a day. So facilitated diffusion is basically when you've got a protein in the membrane that's going to help that polar molecule get through. So it can't go through on its own very well, so you'll have a polar molecule, and typically, you'll have a membrane protein with a polar central core that's being protected from the lipid environment by those transmembrane regions. And so when you do, transport versus concentration gradient, you're going to see a parabolic curve here. And that's because this gets back to Michaelis-Menten, which is all about how fast can that enzyme do its job. So basically, as you go to higher and higher concentration, your transport increases dramatically, and then it starts to level off, and eventually it becomes the same as simple transport here, or simple diffusion. Okay, and the idea is that transporter sitting in the membrane, basically for every molecule that comes in, it has to change conformation once. So it opens to the outside, it grabs a molecule, changes conformation, opens to the inside, the molecule goes in. So it has to go back and forth between those two conformations. And it can only do that so fast. And there are only so many transporters in the membrane. So at a low concentration gradient, you're only activating a few of those transporters. As you increase the concentration gradient, more and more of those transporters get activated, and now your transport rate goes up really, really fast. And then as you get into here, the transporters start to get saturated. So some of them are moving as fast as they can, and eventually you get to a point where they're all moving as fast as they can. And by increasing the concentration gradient, you really can't make them go any faster. They're saturated. And after that point, any transport in is going to be the same as your simple transport, because your transporters are going as fast as they can. Any extra glucose getting in is just going through the membrane. So Michaelis-Menten kinetics then is being followed by these. Okay. And here's more or less how it works. Again, this is from a different textbook, but you've got a number of different transporters. You've got Uniport, and our glucose transporter is like this. There's an amino acid transporter like this. Basically, it's passive. It's not using any energy. Totally reversible. Can go in either direction, and but it's only moving one thing at a time. Then you've got Symport, where it's moving two things at a time. Now, typically, a Symporter is going to be active transport. The reason it's bringing two things at once is because it's going to have one thing going down its concentration gradient in order to bring the other thing up its concentration gradient. So typically, a Symporter is going to be active transport, and we'll talk about that in the next section. And then you've got antiporters, where one molecule goes, or one particle goes in one direction and the other particle goes in the other direction. Now, these can be either active transport or passive transport. For example, we've got in red blood cells, you've got a hydrogen sodium antiporter. And here, the reason you're antiporting is you've got a positive charge and a positive charge. So if you're uniporting hydrogen, positive charges would be leaving. Eventually, you build up negative charges inside the cell, and it wouldn't work anymore because you couldn't lose more positive charges eventually. So in order to keep this thing totally passive, you're letting one positive charge out for another one coming in. We talked earlier about the chloride bicarbonate antiporter. Again, totally passive transporter. But it's antiporting because you've got a negative bicarbonate going out, negative chloride going in, and they cancel each other out. So you're not generating kind of membrane potential with its activity. That helps to keep it passive again. So typically, your Symporter is going to be like a sodium glucose transporter, sometimes a hydrogen something transporter, more common in prokaryotes than eukaryotes, but one is going down its concentration gradient, the other is going up. And glucose, of course, glucose transporter, totally reversible, totally passive. Okay, so that's just kind of general ideas about the types of transporters that we see in cells. Here's a different kind of transporter. It's not really a transporter, it's a membrane, it's a channel. So you might want to click on this later to see that animation. But basically, a channel is a multi-pass protein that's got a water-filled pore in the center of it that allows polar things to go through. And they're both gated and selective. So your channels typically only let one particular particle go through. So you tend to have like a potassium channel, a sodium channel, a chloride channel, a calcium channel. Those are your major channels. They're only going to let that one through. The only exception to that I know of is the nicotinic acetylcholine receptor, which is a cation channel. Sodium or potassium can go through it. Tends to be sodium because of the concentration gradients, but it can go either way. But anyway, selectivity filter, so your channel's only going to let one particular ion go through. And they're typically gated too. There are very few channels that aren't gated. We'll talk about leak potassium channels later today. They're not gated, but most of them are going to be gated. So they're not always open. They're going to be closed, and when they're stimulated to open, they open. And when they do, ions come flying through much, much faster than a transporter like this. With these transporters, it has to change conformation for every particle going through. With these, they have one conformational change, and then the ions come flying through. Oops, ooh, what did I do? Here's kind of what they actually look like. They look more like this. You can see this from the top view. You can see the water-filled pore in the center, and these 5 little alpha helices here are, testing to see what ions going through. That's your selectivity filter. And again, check this out later and see what that looks like. Here is a water channel. Your book stresses this for some reason. Water channels, one place you're going to see them is in the collecting ducts of the tubules, in the collecting ducts of the kidneys. And you will, oh, I'm sorry. If the kidneys want to lose more water, they'll put more water channels into the membrane and more water will go out. You'll have a more dilute urine. If they want to conserve water, they'll come back out of the membrane. And that movement of those water channels in and out of the collecting ducts is all controlled by hormones. One of the most important one is ADH, antidiuretic hormone, will control the movement of those water channels. But again, water can go directly through the membrane, but if you've got a channel, it's going to go through a lot faster. So this helps water to move in and out a lot faster. Okay. I believe, okay. Do I go on here? Let's see. Okay, no, this is pumps. Nope. Okay, there are a lot of different types of channels. I don't have a slide for this, but I have it in your notes. And different kinds has to do with different selectivities and also different types of gating. Later in this chapter, we'll talk about voltage gated channels, which are channels that will open when the membrane voltage changes. We've also got ligand gated channels, which are going to be activated by hormones. You'll see them a lot in synapses. So a neurotransmitter is released and it binds to a channel and the channel opens. So that's a different type of gating. You've got stretch activated channels, which you see them in a lot of places, but especially in sensory neurons, like your touch channels, your Purkinjean corpuscles or whatever. When you touch something, your cell membranes get deformed and stretched, ions go through, and that activates an action potential. So stretch channels are important for sensory neurons. They're also important in smooth muscle. If your blood pressure goes up, your smooth muscles get stretched, and that can cause them to contract because you'll get stretch activated channels that allow sodium to come in, depolarizing, and then that causes a contraction of the smooth muscle. So you've got stretch activated channels in a lot of different places. Cyclic nucleotide gated channels. I think the best example I can think of is in your retina. in the retinal cells, you've got, I think they're cyclic GMP activated sodium channels. And the weird thing about retinal cells is they're like backwards of what you would think. You would think that if light came in, it would instigate an action potential, but actually cyclic nucleotides are opening those sodium channels until light comes in, and then they close, and that's what signals a photon has entered. So that's not important now. I'm just giving you an example, but there are channels that are also gated by cyclic nucleotides. And there are other gatings as well, but those are the ones that we'll probably talk about later in this class. So water channels we see in the kidney a lot, and also porins, which are, more, porins are smaller and they're more prokaryotic. So we're going to see them in mitochondria and chloroplasts in our eukaryotic cells. Okay, so I think you people are probably tired of listening to me talk at this point. I'm getting a little bit hungry. So that's about all I want to do today. I'm hoping that I recorded this. I tried to. Again, I'm still kind of learning the ropes here in this new software environment. But that's all I have for today. Do you have any questions before I close this chat or video? Any questions? Well, thank you. I'll try to have a good day.
Thank you.
Oh, you're welcome. All right, we're gonna